Dear listener, hello, and welcome to the Deep Sleep Science channel. Tonight, we'll drift gently into the deepest parts of the ocean, where sunlight never arrives, and where life learns to live by different rules. In those quiet, pressurized depths, even a small glimmer can become a signal, a disguise, or a trap. And in the steady darkness, bodies, senses, and entire ecosystems reshape themselves around scarcity, chemistry, and patience. Black sea devil in the genus Melanostus is a deep-sea anglerfish that lives where sunlight never reaches, and it turns that darkness into an advantage with a bioluminescent lure called an escal. In the black midwater, where prey may be separated by long distances, that tiny light becomes the most noticeable thing for meters around, drawing curious animals close enough for a sudden ambush with long, inward, curving teeth. This animal also reflects how reproduction changes in vast, dark habitats where encounters are rare. Many anglerfish lineages show extreme sexual dimorphism, with tiny males that locate females and may fuse to them, ensuring that reproduction can happen even when finding a partner is difficult in open, lightless water. The body plan, the trap-like jaws, the energy-saving lifestyle, and the controlled signal of the lure all assume a world where the sun never appears. The vampire squid, Vampiroteuthis infernalis, lives in deep conditions where oxygen can be scarce and light is nearly absent, and it is not a true squid, but the sole living representative of its own order. Rather than relying on speed, it uses an unusually efficient metabolism suited to low oxygen layers, and it carries bioluminescent organs that can help confuse predators instead of lighting up the water like a lantern. Its feeding strategy is especially adapted to a dark, low-energy life. Instead of actively chasing prey, it can collect drifting organic particles, often called marine snow, using long filaments and pass them toward the mouth, making a quiet living on the steady rain of debris from above. It is a creature designed for stability, dark water, thin oxygen, and food that arrives slowly and unpredictably. The barrel-eye fish, Macropina microstoma, lives deep enough that sunlight is essentially gone, with only the faintest glow and the sudden flickers of bioluminescence remaining. It carries a transparent dome over its head that shields extremely light-sensitive tubular eyes, which are oriented upward to detect tiny glimmers and silhouettes above in the dim water. One of its most surprising features is that what look like eyes at the front of the face are actually olfactory organs, while the true eyes can pivot to look forward when the fish closes in on prey. This design is less like ordinary seeing and more like precision detection of the most meaningful light available at depth, bioluminescent signals from other organisms, faint and brief, but rich with information. Praia dubia is a giant siphonophore, a colonial predator that lives in deep mid-water where sunlight is absent, and it behaves like a living net. Rather than being a single-bodied animal, it is a colony of specialized units called zooids that function together as one, deploying long curtains of tentacles armed with stinging cells to snare prey that drifts into cabala. Contact. Because it is modular, it can combine parts that do different jobs: feeding, movement, and reproduction more like a cooperative machine than a single organism. In the deep sea, where prey can be sparse and swimming constantly can be expensive, this approach can be efficient, maximize capture area, reduce wasted motion, and let the ocean's slow drift deliver opportunities. The aphotic zone is the region of the ocean where sunlight does not penetrate, spanning the bathypelagic and deeper waters and representing a vast portion of Earth's habitable volume. In this setting, animals cannot rely on photosynthesis-driven food webs locally, so they depend on falling organic matter, predation chains built around that sinking food, all chemosynthesis-based systems that draw energy from chemicals instead of light. This environment shapes a familiar set of traits across many groups. Reduced or absent pigmentation, huge mouths relative to body size, pressure-tolerant tissues, and sensory systems tuned to vibration, chemicals, and bioluminescent cues rather than daylight vision. In many cases, seeing becomes less about color and detail and more about detecting the faintest contrast or the briefest flash. Abyssal trenches are Earth's deepest permanently dark habitats, generally below about 6,000 meters, where the water is near freezing and the pressure is enormous. Life persists there, but it is often highly specialized, with many species restricted to particular trenches and adapted to scarce, patchy food supply in an environment that is both vast and physically extreme. Trenches can also function as collection points where organic material accumulates, creating benthic communities supported by what the deep topography gathers. Even so, the food remains limited and uneven, which favors efficient scavengers, slow-growing species, and animals that rely heavily on chemical sensing to locate scattered, bait-like resources across the Prosthear. Seafloor. Adult snailfishes in the family Liparidae have achieved notable success in trenches, and in some systems, they are among the most colon fishes recorded. Their bodies are typically soft and gelatinous, a trait that can be advantageous under extreme pressure, where rigid structures can be costly or fragile, and where neutral buoyancy and flexibility can matter more than yaw. Armor. They also show that deep-sea dominance does not require a single monster blueprint. Some of the most successful hadelfishes are not heavily armored predators. They are pressure-adapted, energy-efficient swimmers that exploit available prey such as crustaceans and other trench fauna. Their success suggests that fitting the physics and chemistry of extreme depth can be as important as weaponry. Riftia pachytilla is a hydrothermal vent tube worm that can grow large near black smoke vents, and as an adult, it lacks a digestive tract entirely. Instead, it relies on internal symbiotic bacteria that convert vent chemicals into usable energy, using chemosynthesis rather than photosynthesis, and turning a lightless environment into a place where biomass can be surprisingly dense. This lifestyle is a direct answer to a world that never sees the sun. The tube worm supplies its bacteria with chemicals such as sulfides and with oxygen via specialized tissues and blood chemistry, effectively transforming a toxic neighborhood into a high-productivity oasis. In this way, the worm becomes a living support system for an internal microbial engine powered by Earth's chemistry. Hydrothermal vent ecosystems are animal communities powered by chemicals, and they can support dense life in deep regions that are otherwise food poor. Instead of relying mainly on marine snow from above, these systems are fueled by chemosynthetic microbes that use chemicals from vent fluids to build organic matter, forming the base of a distinct food web that functions without sunlight. The result is a deep-sea paradox. Total darkness can still host high biomass and fast growth, but only where geology supplies concentrated chemical energy. This creates patchy, island-like ecosystems, clusters of life around vents separated by large stretches of comparatively barren seafloor, where the location of chemical gradients becomes the map that defines a NAP, where entire communities can exist. Bioluminescent signaling is the deep sea's substitute for sunlight, and in aphotic waters, many organisms generate light through biochemical reactions. Unlike sunlight, which is external and broad, bioluminescence is controlled, local, and information-rich, used to lure prey, confuse predators, communicate, or provide camouflage through counter-illumination. In this world, light becomes less like illumination and more like language. A glow, pulse, or flash can signal identity, direction, or danger, and the ability to read these cues becomes as important as the ability to produce them. The deep sea is not a place of no light, so much as a place where light is rare, purposeful, and often alive. The pelican eel, Eurypharynx pelicanoides, lives in deep midwater where sunlight is absent and prey encounters can be unpredictable. Its most striking feature is an enormous, bag-like mouth that can open extremely wide compared to its body, allowing it to engulf prey that might otherwise be too large or too agile to catch with precise bites, turning a brief encounter into a meal by swallowing first and sorting later. This design reflects deep-sea energy economics, where meals can be scarce and widely spaced. A large, expandable catch net mouth can make sense when opportunities are rare, and the eel's overall build supports low-energy drifting with short bursts of motion when needed. It is a body plan shaped for a life of patience, punctuated by decisive moments. Gulper eels, in the genus Sacchopharynx, occupy very deep water where ambient light is effectively zero, and their feeding design is an extreme version of take what you can get. Their jaws and throat tissues can expand dramatically, allowing them to swallow prey nearly as large as themselves, converting rare opportunities into substantial caloric returns. Their bodies are typically long and slender, minimizing the energetic cost of moving through vast volumes of dark water. Like many deep-sea fishes, they prioritize efficiency over speed, relying on specialized anatomy, expandable jaws, flexible skull elements, and stretchable stomachs when the rare moment for feeding arrives. Viperfishes in the genus Scholliodus inhabit deep zones where sunlight is absent and combine formidable teeth with light-producing organs. Their long, needle-like teeth can interlock outside the mouth, forming a cage that traps prey, and their bioluminescent photophores can support signaling, lure-like attraction, or camouflage strategies that Tabribar influence how other animals perceive them. In the deep ocean, a small glow can be mistaken for food, a mate, or harmless background. Sparkle-so bioluminescence can function as deception as much as visibility. Viperfish thrive by merging two deep-sea tactics, making contact decisive through teeth and jaws, and shaping the information field through controlled light. The stoplight Lucijaw, Malacosteus niger, is associated with a rare sensory trick in the deep sea. It can produce and detect longer wavelength red light in an environment where most animals are tuned to blue-green. Because blue-green travels farther in seawater and dominates most bioluminescence, red can function like a private illumination system, revealing prey without advertising itself broadly to nearby Timah animals. Its jaw system is also specialized for capturing prey efficiently in darkness, where chances can be brief. The deeper lesson is that, in a sunless world, even small differences in what wavelengths you can perceive can create major hunting advantages. A predator that can see with a light others cannot detect may be able to approach more closely and strike with less warning. The Dumbo octopus in the genus Crimpoteuthis lives in deep zones where sunlight does not reach, often hovering just above the seafloor or moving slowly along it. Its ear-like fins provide efficient, low-energy propulsion compared with constant jetting, which would be costly in cold, food-limited depths, making it well-suited to a life where conserving calories is part of survival. Its feeding tends to focus on small benthic animals such as crustaceans and worms, taken from the seafloor rather than chased in open water. In permanent darkness, it relies more on touch and chemical sensing than on vision, and its gentle, energy-saving movement reflects a habitat where patience and efficiency can matter as much as speed. Atolla jellyfish, Atolla wyverlei, inhabits deep midwater where sunlight is absent and the visual world is dominated by bioluminescent flashes. It is known for producing distinctive flashing patterns when attacked, a response that is not meant for navigation, but for changing the predator-prey interaction in a place where a sudden bright signal stands out sharply. against near total black. This is often described as a burglar alarm strategy. By flashing, the jelly may attract a larger predator that could interfere with or deter the attacker. In a habitat without sunlight, defensive light can be highly conspicuous and can shift risk away from the jelly. It is a quiet reminder that, in the deep ocean, light often functions as influence and information rather than as a way to reveal scenery. Some deep-sea fishes are described as ultra-black because their skin absorbs exceptionally high proportions of light, including bioluminescent light emitted by other organisms. In the deep ocean, where the dominant light sources are tiny flashes rather than sunlight, absorbing rather than reflecting can be an effective camouflage, since even a small reflection could reveal a predator's outline during an ED approach. Ultra-black surfaces can be achieved through microstructures in the skin combined with pigmentation, reducing backscatter and stray reflections. In a habitat with no daylight, minimizing reflective signatures can matter more than matching any background color pattern. It is camouflage tuned to a world of point light signals, where the goal is to disappear, not into brightness, but into the absence of it. Xenophyophores are unusually large single-celled organisms, protists found in deep-sea settings including trench regions, and they build complex, fragile structures by cementing sediment grains and particles together. Even though they are technically one cell, they can form sizable frameworks on the seafloor, influencing the small-scale structure of benthic communities in permanent darkness. Their success depends on detrital food and persistence where energy is low, and their ecological significance comes from the habitat they create. By increasing seafloor complexity, they provide micro-refuges and attachment surfaces for other organisms, demonstrating how even microscopic life can become an architect in the deep ocean's quiet landscapes. Bone-eating worms in the genus Osedax specialize in colonizing bones, especially from whale falls, in deep, dark waters where sunlight never reaches. They lack traditional mouths and instead use root-like structures that penetrate bone, often supported by symbiotic bacteria that help access nutrients, letting them exploit an energy source that is rare but extraordinarily rich when it arrives on the seafloor. Whale falls create extended ecological successions, from scavengers consuming soft tissue to microbes and specialists exploiting fats and compounds over time. Osedax represents an extreme form of specialization for capturing that energy over years, turning what might look like lifeless bone into a long-term habitat and food source in an otherwise resource-limited abyss. Whale fall communities are deep-sea hotspots where a single carcass can support a complex community for extended periods in total darkness. Early stages involve scavengers rapidly removing soft tissues, while later stages are shaped by microbial activity and specialized invertebrates that exploit remaining fats and bone-derived resources, creating a temporary reef-like structure in areas that may otherwise be sparsely populated. These systems show how deep-sea ecosystems can be transformed by rare, massive subsidies from surface life. Even without any local photosynthesis, the deep-sea floor can experience bursts of biodiversity and biomass when a large carcass arrives, reshaping local food webs and supporting specialists that may be encountered only when such events occur. The scaly foot snail is often highlighted as a remarkable hydrothermal vent endemic from the Indian Ocean, reflecting how isolated vent fields can produce unusual, locally adapted species in total darkness. Its presence points to the strong influence of vent chemistry on evolution, with animals often tightly matched to the conditions and resources provided by particular vent systems. More broadly, it teaches a lesson in biogeography. Vent habitats are patchy and separated, and different oceans or ridges can host distinct species. assemblages because connectivity is limited and dispersal is challenging. Never seeing light is the shared condition, but local geology, chemical regimes, and the difficulty of colonizing new vents shape which evolutionary solutions arise and persist. Yeti crabs, in the genus Kiwa, live in deep-sea habitats often associated with hydrothermal vents or related chemical settings, where sunlight is absent. The environment is shaped by flows of reduced chemicals. Some species have dense CT hair-like structures that host microbial communities, and the crabs can position themselves in chemical flows that feed microbial growth, then harvest the microbes as food. This is a striking adaptation to a sunless world. Rather than searching widely for prey, an animal can cultivate a food source directly on its own body in a chemically enriched zone. It is a localized survival strategy that ties nutrition to geochemical flux, offering a different route from detritus dependence while still functioning entirely without sunlight. Large-scale darkness defines most of the living space on Earth because the bathypelagic, abyssopelagic, and hadal zones together make up the majority of the pref. Ocean's habitable volume. These regions never receive sunlight, not even as a faint glow, and yet they host countless organisms whose entire evolutionary history unfolds, without day, night, or seasons driven by the sun. In this immense dark water, ecological rules shift away from visual dominance. Energy flows slowly, distances are vast, and encounters between organisms can be rare. As a result, life here is shaped less by competition for light and more by efficiency, patience, and the ability to interpret subtle signals carried by chemistry, vibration, and occasional flashes of living light. Hadal trenches represent the deepest large-scale environments on the planet, stretching downward into zones of crushing pressure and complete darkness. These trenches are not continuous, but scattered across the ocean floor, forming isolated basins that function like biological islands separated by quay, ridges, and abyssal plains. Because of this isolation, each trench can develop its own biological character. Species may be restricted to a single trench system, evolving independently under the same absence of light but slightly different food inputs, sediment, properties, and water chemistry. Darkness is constant, but geography quietly shapes diversity. Across the Arctic Ocean, energy does not originate locally from sunlight, but arrives indirectly, delayed and transformed. Organic matter produced at the surface sinks slowly downward as marine snow, losing mass as it falls, until only a fraction reaches the deep sea floor where it becomes the foundation of entire ecosystems. This delayed delivery creates a time-shifted connection between surface life and deep ecosystems. A bloom at the surface may influence deep communities weeks or months later, even though the organisms living there will never experience the light that made that bloom possible. Hydrothermal vents introduce an entirely different large-scale pattern by breaking the dependence on surface-derived energy. Here, geological processes bring chemical-rich fluids from Earth's interior, allowing microbes to produce organic matter directly in complete darkness. These vent systems form biological oases scattered across otherwise food-poor seafloor, creating sharp contrasts between dense life at vents and sparse communities only a short distance away. On a planetary scale, vents demonstrate that sunlight is not the only engine capable of sustaining complex ecosystems. Cold seeps add another layer to the deep ocean's large-scale structure, releasing methane and sulfide from sediments rather than from volcanic heat. Though cooler and often more stable than vents, they still support rich communities powered entirely by chemical energy instead of light. Over long timescales, seeps can persist for decades or longer, allowing extensive biological structures to form. In a sunless ocean, these chemically driven systems act as long-term anchors of productivity, quietly reshaping surrounding seafloor ecology. Whale falls represent massive but rare energy injections into deep ecosystems, delivering tens of tons of organic matter from the surface to the abyss in a single event. In a landscape defined by scarcity, such falls become ecological milestones rather than ordinary occurrences. Their influence extends far beyond the immediate area, supporting successional communities that can last for years and connecting deep species across distances through dispersal. Even in permanent darkness, life responds strongly to rare pulses of abundance. On abyssal plains, which cover enormous areas of the seafloor, life appears sparse, but is governed by subtle, large-scale patterns of food deposition, currents, topography, and sinking particle pathways, creating visible landscapes of slightly richer and poorer zones. Animals living here navigate a world shaped by gradients rather than landmarks. Without light, differences in sediment chemistry or organic content become the defining features that guide where communities form and persist: sponges, corals, and associated animals. At a large scale, seamounts act as biological stepping stones, connecting populations across deep basins and increasing overall diversity. In a world without light, physical structure becomes a primary driver of ecological complexity. Deep-sea coral gardens illustrate how reef-like ecosystems can exist entirely without photosynthesis. These corals capture passing particles from currents, slowly building complex frameworks over centuries in cold, dark water. On a planetary level, such habitats challenge the idea that reefs require sunlight. Instead, they show that structural ecosystems can arise wherever energy flow, time, and stability align, even in perpetual night. Glass sponge reefs extend this idea further, forming rigid, long-lasting frameworks in deep, dark waters. Their silica skeletons persist for centuries, creating stable habitats that accumulate biodiversity over time. These reefs demonstrate that longevity itself is a large-scale ecological force in the deep sea. When change is slow and light is absent, ecosystems can be shaped by structures that endure rather than by rapid turnover. Trenches function as large-scale collectors of organic matter, with their steep walls and depths trapping sinking debris that might otherwise disperse across plains. This accumulation can elevate food availability relative to surrounding areas. As a result, trench ecosystems often differ markedly from adjacent abyssal zones, even though both are equally dark. Depth and shape, rather than light, become the defining environmental variables. Across all these environments, darkness removes the daily and seasonal rhythms that structure surface ecosystems. There is no sunrise or sunset, no seasonal bloom triggered by increasing light, and no visual cues tied to time. This absence of rhythm leads to ecological stability punctuated by rare events. Instead of frequent cycles, deep ecosystems experience long, quiet periods interrupted by pulses, food falls, vent eruptions, or shifts in currents. The deep ocean also represents one of Earth's largest carbon sinks, with organic material buried in sediments for long periods. Animals influence this process through feeding, burrowing, and sediment mixing, shaping long-term carbon storage. Even without sunlight, deep ecosystems play a global role in regulating Earth's carbon balance. Life that has never seen light still quietly influences planetary chemistry. From a planetary perspective, the deep ocean shows that most of Earth's biosphere exists beyond human sight, operating under rules shaped by pressure, chemistry, and time rather than by light. Deep ocean did not appear suddenly, but unfolded slowly as animals followed food and opportunity downward over evolutionary time. As surface-produced organic matter began reaching deeper layers, Some lineages gradually adapted to colder temperatures, higher pressure, and diminishing light, until sunlight disappeared entirely from their world. This descent into darkness reshaped bodies, senses, and life histories. Over millions of years, evolution favored traits that worked without visual cues, allowing entire branches of the tree of life to settle permanently in places where the sun has never shone. Many deep-sea fish lineages trace their origins to shallower ancestors that once relied on vision and faster swimming. As populations moved deeper, eyes often became larger and more light-sensitive at first, then in some cases reduced again as even bioluminescent. Hues became scarce and costly to process. This evolutionary path shows that adaptation to darkness is not linear. Some species enhance vision to its physical limits, while others abandon it almost entirely, replacing sight with smell, touch, and vibration, sensing that work reliably in absolute blackness. Bioluminescence itself is an ancient evolutionary innovation that became increasingly important as animals moved deeper. What may have begun as a simple biochemical byproduct evolved into controlled light production used for communication, predation, camouflage, and defense. Over time, light-producing organs became more specialized and precisely regulated. In the absence of sunlight, evolution turned light into a tool rather than an environmental condition, shaping interactions in ways that have no direct parallel in B. Surface ecosystems. The rise of chemosynthetic ecosystems marked a major evolutionary shift in deep-sea history. When life colonized hydrothermal vents and cold seeps, it broke free from total dependence on surface-derived energy, tapping directly into Earth's internal chemistry. This allowed the evolution of large, dense communities in complete darkness, supported by symbiosis between animals and microbes. These systems represent one of the most profound departures from sunlight-based life on the planet. Symbiotic relationships became especially important in the deep sea, where hosting energy-producing microbes could mean the difference between scarcity and abundance. Tube worms, clams, and mussels evolved internal structures to house bacteria that convert chemicals into organic matter at the cellular level as a response to darkness. Reproductive strategies also shifted as species adapted to deeper environments. With fewer encounters between individuals, some animals evolved extreme solutions, such as reduced males, chemical mate detection, or long-term brooding of offspring. These strategies increased the likelihood of successful reproduction in vast, sparsely populated habitats. Evolution in darkness rewarded certainty and efficiency over speed and quantity. The development of soft, gelatinous bodies in many deep-sea animals reflects long-term evolutionary trade-offs. In cold, dark environments with limited food, reducing the energetic cost of building tissue became more important than maintaining rigid strength. Gelatinous forms also tolerate pressure well and often provide poor caloric reward to predators, indirectly increasing survival. Over evolutionary time, softness became a viable and even advantageous solution to life without light. Gigantism in certain deep-sea crustaceans and invertebrates represents another evolutionary experiment. Larger size can improve energy storage, mobility across long distances, and success in scavenging rare food falls. At the same time, other lineages evolved toward miniaturization, reducing energy needs and exploiting microscopic food sources. Darkness did not favor one size, but rather amplified the importance of matching body scale to feeding strategy. Eye structure evolved in diverse ways across deep-sea lineages. Some species developed tubular eyes, optimized for detecting faint points of light above, while others retained wide-angle eyes to sense flashes from any direction. In the deepest habitats, where even bioluminescence is rare, eyes may become vestigial or disappear altogether. Evolution favors usefulness, and in absolute darkness, vision can become optional. Skin pigmentation also changed as animals adapted to lightless conditions. Deep reds, browns, and ultra-black surfaces evolved because they absorb or hide stray light rather than reflect it. These pigments do not serve aesthetic or signaling roles as they might at the surface. Instead, they function as stealth technology, shaped by evolutionary pressure to remain invisible in a world defined by darkness. Long lifespans emerged as a common evolutionary outcome in deep habitats. Cold temperatures slow metabolism, and limited food discourages rapid growth, favoring species that spread survival and reproduction across decades or longer, patience and ambush. Many predators evolved to wait motionless for long periods, relying on sudden decisive strikes rather than active pursuit. In a world without light, movement itself can be costly and revealing. Over time, stillness became a successful evolutionary strategy. The evolution of chemical sensing became increasingly refined as vision lost dominance. Olfactory organs, taste receptors, and chemical processing brains expanded in importance, allowing animals to follow invisible trails of food and mates. These sensory changes illustrate how darkness reshaped perception itself. Evolution replaced sight-based awareness with a chemical map of the environment. Pressure tolerance required deep biochemical evolution. Proteins, enzymes, and cell membranes evolved structures that remained stable under crushing forces that would disable shallow water organisms. These molecular adaptations are among the most fundamental evolutionary changes, allowing life to function at depths once thought uninhabitable. Trench ecosystems represent some of the most isolated evolutionary arenas on Earth. Separated by geography and pressure barriers, trench populations diverged independently, producing unique species adapted to very narrow conditions. Evolution in these isolated basins mirrors island evolution on land, but with darkness and pressure replacing distance and climate as the dominant forces. Over geological time, the deep ocean remained relatively stable compared to surface environments. This stability allowed ancient lineages to persist with fewer disruptions, preserving evolutionary designs that might have vanished elsewhere. As a result, the deep sea hosts a mixture of ancient traits and highly specialized innovations, all shaped by life without light. The evolutionary history of deep-sea life shows that darkness is not merely an absence, but a powerful selective force. It removes visual shortcuts, slows time, and rewards efficiency, cooperation, and subtlety. Through this long process, life learned not just to endure darkness, but to build entire worlds within it. In the deep ocean, structure and environment replace sunlight as the main forces shaping how ecosystems function. Temperature, pressure, chemistry, and slow-moving water together create a stable but demanding world, where even small environmental gradients can determine where life gathers and how it survives, steadily with depth, becoming one of the most defining structural forces in deep-sea environments. Every additional meter adds weight, compressing tissues and altering how molecules behave. Deep-sea organisms are built around this reality, with bodies and cellular structures designed to function normally under forces that would crush surface-dwelling life. This pressure gradient effectively divides the ocean into stacked biological zones, even where light is absent everywhere. Species distributions often align more closely with pressure tolerance than with depth alone, creating invisible boundaries that structure deep ecosystems. Temperature in the deep ocean is remarkably stable, remaining cold and nearly constant year-round. Without sunlight to warm surface waters or create seasonal variation, deep habitats experience a steady thermal environment that strongly influences metabolism, growth, and development. This thermal stability allows organisms to finely tune their physiology to narrow temperature ranges. At the same time, it discourages rapid growth and favors slow, energy-efficient life strategies that match the calm but resource-limited conditions. Ocean currents shape deep ecosystems by controlling the delivery of food particles and chemicals. Even weak currents can be critically important, transporting marine snow, dissolved compounds, and larvae across vast dark spaces. Where currents interact with seafloor features like ridges or slopes, food can become concentrated, creating productive zones in otherwise sparse environments. In darkness, water movement becomes a primary organizing force. Seafloor topography adds structure to deep habitats, creating slopes, basins, ridges, and trenches that influence sediment accumulation and flow patterns. These physical features guide where organic matter settles and where animals can most efficiently feed. A trench may collect sinking debris, while a ridge may deflect currents upward, shaping entirely different biological communities despite sharing the same absence of light. Sediments themselves form layered environments, with oxygen-rich surfaces transitioning to low-oxygen zones just beneath. These chemical gradients shape where burrowing organisms can live and how deep they can penetrate into the seafloor. For animals living within sediment, darkness is absolute, and chemistry replaces vision entirely. The structure of mud and the availability of oxygen become the defining environmental features. Hydrothermal vents introduce extreme structural contrasts into deep environments, releasing hot, chemically rich fluid through cracks in the seafloor. These vents create steep temperature and chemical gradients over very short distances. Itself becomes a map for life in total darkness. Cold seeps function differently, releasing chemicals more slowly and often over broader areas. Their relative stability allows long-lived communities to develop, anchored by organisms that rely on chemical energy rather than sunlight. The seep structure alters sediment chemistry over time, creating persistent habitats that support complex biological interactions without visual bark. Cues. Biological structures built by animals play a major role in shaping deep ecosystems. Tubeworm clusters, mussel beds, coral frameworks, and sponge reefs add vertical relief to otherwise flat seafloor landscapes. These structures change water flow, trap particles, and create shelter, multiplying the number of available niches in a dark environment where physical breadth, complexity is otherwise limited. Glass sponge reefs exemplify long-term structural stability in deep water. Their silica skeletons resist decay and can persist for centuries, forming rigid frameworks that support diverse communities. In darkness, where change is slow, such durable structures become ecological anchors, shaping local biodiversity over geological timescales. Deep-sea coral structures similarly add complexity, growing slowly but steadily as they filter particles from passing currents. Their branches provide attachment points and refuges for many other organisms. Even without photosynthesis, these corals demonstrate how structure alone can sustain rich communities in lightless environments. Chemical gradients near vents and seeps create sharp environmental boundaries that structure ecosystems more strongly than light ever could. Small shifts in chemistry can mark the difference between habitable and lethal conditions. Animals living in these zones must constantly balance access to energy with tolerance to toxicity, positioning themselves precisely within invisible chemical landscapes. Oxygen availability varies across deep habitats, influencing where species can survive. Some regions, such as oxygen minimum layers, restrict which animals can function effectively. Species adapted to low oxygen gain access to ecological space with reduced competition and predation, using chemical and physiological tolerance as structural advantages. Particle size and composition influence feeding strategies across the deep ocean. Fine particles favor deposit feeders and filter feeders, while larger debris support scavengers and predators. These physical properties of sinking material help define which feeding strategies dominate in different dark environments. Animal movement and even distant surface processes can be detected. For some organisms, these acoustic cues provide information about habitat, danger, or opportunity in the absence of visual landmarks. Light introduced by humans during exploration temporarily alters deep environments. Artificial illumination can attract, repel, or disorient animals that evolved in perpetual darkness. This highlights how sensitive deep systems are to structural changes, even brief ones, and how darkness itself is a defining environmental condition. Over long timescales, the deep ocean remains one of the most stable environments on Earth. Without sunlight-driven cycles, change occurs slowly, shaped by tectonics, sedimentation, and chemistry. This stability allows ecosystems to persist quietly, structured by forces that act over centuries rather than days. In these environments, structure replaces spectacle. Subtle gradients, slow flows, and enduring frameworks guide life through darkness. The deep sea becomes a place where the environment speaks softly and life listens carefully. In the deep ocean, interactions unfold slowly and quietly, shaped by mechanisms that operate at medium scales between whole ecosystems. Microscopic chemistry. These processes govern how energy moves, how organisms interact, and how life maintains balance in a world without sunlight. What follows are the steady, underlying interactions that allow deep-sea systems to function, even when nothing seems to be happening at the surface. Energy transfer in the deep sea often depends on trophic coupling, where material produced in one part of the ocean is consumed far below. Sinking organic particles are intercepted by midwater organisms, transformed through digestion, and passed onward as waste or prey, gradually stepping energy downward through the food web. This layered transfer creates a vertical chain of dependence. Each level relies on what survives from the one above, making efficiency and recycling essential in the absence of continuous energy input. Predator-prey interactions in darkness emphasize surprise and proximity rather than speed. Many predators rely on ambush or interception, positioning themselves where prey is likely to pass, and striking only when contact is nearly guaranteed. This interaction style reduces wasted energy and reflects the cost of motion in cold, food-limited environments. Success depends less on pursuit and more on timing and placement. Scavenging plays a central role in deep ecosystems, connecting rare, large food falls to broader community nourishment. When a carcass arrives, scavengers rapidly consume and fragment it, converting concentrated biomass into dispersed nutrients. These interactions ensure that energy from a single event spreads through multiple trophic levels. Scavengers act as mediators, redistributing value across the ecosystem rather than allowing it to remain locked in one location. Chemical signaling governs many deep-sea interactions, replacing visual communication. Dissolved molecules released by prey, decaying matter, or potential mates create gradients that guide movement and behavior. Following these gradients requires integration of time and space as signals disperse and shift with currents. Successful navigation depends on sensitivity and patience rather than direct pursuit. Competition in the deep sea is often subtle and prolonged rather than immediate and aggressive. Limited food availability means organisms rarely encounter each other directly, but they still compete through efficiency and positioning. Those that can exploit resources slightly better by digesting more thoroughly or detecting cues more quickly gain long-term advantages that shape population structure over time. Symbiotic interactions form a major mechanism for survival in dark environments. Animals hosting chemosynthetic bacteria gain direct access to energy sources unavailable to others, effectively bypassing detritus-based limitations. These partnerships blur the line between organism and ecosystem, with energy production occurring inside living bodies rather than in the environment at large. Filter feeding acts as a stabilizing mechanism in deep systems, removing particles from the water and converting diffuse energy into biomass. This process smooths fluctuations in food availability, providing steady nourishment where large prey is rare. By filtering continuously, these organisms help regulate particle loads and influence microbial populations, subtly shaping surrounding conditions. Deposit feeding similarly stabilizes energy flow by extracting value from sediments over long periods. Rather than relying on pulses, deposit feeders exploit widespread, low-quality resources. Their constant processing of sediment helps recycle nutrients and maintain ecosystem function even when new input is minimal. Bioluminescent signaling introduces an interaction mechanism unique to darkness. Light pulses can attract, repel, or mislead, altering predator-prey dynamics in ways that have no equivalent in sunlit environments. These signals are brief and controlled, making interactions discrete rather than continuous. Each flash becomes a moment of information exchange. Counter-illumination functions as an interaction between organism and observer, rather than between two organisms. By matching background brightness, animals manipulate how others perceive them. This interaction relies on geometry and timing, reducing detection rather than confrontation. It is a defensive mechanism embedded in perception itself. Reproductive interactions are shaped by low encounter rates. Many species rely on chemical cues or long-lived gametes to increase the chance of fertilization. Some invest heavily in fewer offspring, shifting interaction from quantity-based reproduction to quality-based survival. Larval dispersal acts as a delayed interaction between generations. Larvae released into deep currents may travel long distances before settling, connecting distant populations. This process spreads genetic material slowly and unpredictably, reinforcing isolation while still allowing occasional exchange. Parental care modifies survival interactions by reducing early life mortality. In deep environments, protecting offspring can be more effective than producing many vulnerable young. This shifts interaction from environment versus offspring to parent versus risk, redistributing energy investment across the life cycle. Predation pressure in the deep sea is often low frequency but high impact. Encounters are rare, but outcomes are decisive. This shapes behavior toward caution and efficiency, minimizing unnecessary exposure while maximizing success when interaction occurs. Burrowing interactions alter sediment structure and chemistry. As animals move through mud, they oxygenate layers, redistribute organic matter, and create microhabitats. These interactions affect not only the burrower, but also surrounding microbial and invertebrate communities. Microbial mediation underlies many interactions indirectly. Microbes process organic matter, release chemical cues, and form the base of food webs that larger organisms depend on. Though invisible, microbial activity drives most deep-sea interactions from the bottom up. Competition between this diversity of mechanisms allows ecosystems to persist despite low overall energy availability. Movement interactions are minimized to conserve energy. Many animals reduce locomotion, interacting with their environment through sensing rather than roaming. This stillness shapes how interactions unfold, favoring interception over chase. Temporal interactions dominate over immediate ones. Effects accumulate slowly, and small advantages compound across years. Deep ecosystems are shaped by long-term balances rather than rapid shifts. Together, these mechanisms form a quiet web of interactions that sustain life in darkness. Each process is gentle, deliberate, and persistent. Nothing rushes. Nothing is wasted. Everything flows slowly onward. At the smallest scales of the deep ocean, life continues its work quietly, shaped by particles, molecules, and microscopic interactions that unfold far beyond the reach of light. These fine-scale processes are slow, persistent, and essential, forming the hidden foundation that allows larger organisms and entire ecosystems exist in permanent darkness. Here, nothing is hurried. Chemistry, physics, and biology blend together in subtle ways, sustaining life one particle at a time. At the microscopic level, marine snow is not a single substance, but a complex mixture of dead plankton, fecal pellets, mucus, and fragmented organic debris. As it sinks through dark water, it is continuously altered by bacteria and tiny grazers, losing mass and changing composition before it ever reaches the par. Seafloor. For deep-sea life, this transformation matters. The nutritional value, size, and density of particles determine which organisms can use them and how efficiently energy is transferred. What arrives in the abyss is not raw surface production, but a carefully filtered remainder shaped by countless microscopic interactions along the way. Bacteria play a central role in deep-sea ecosystems by breaking down organic material into simpler compounds. In the absence of sunlight, bacterial metabolism becomes the primary engine of recycling, converting complex molecules into forms usable by other organisms. These microbes operate continuously, coating particles, sediments, and biological surfaces. Their activity releases nutrients, produces chemical cues, and supports higher trophic levels, making them indispensable architects of deep-sea energy. Flow extremely low. At the smallest scales, these organisms quietly expand the range of environments that can support living systems without light. At the particle level, aggregation and disaggregation constantly reshape food availability. Tiny particles collide and stick together, forming larger aggregates that sink faster, while turbulence and grazing break them apart again. These processes control how fast energy moves downward and who can access it. For deep-sea organisms, the size of a particle can determine whether it becomes food, sediment, or something lost forever to burial. Biofilms form on nearly every available surface in the deep ocean, from rocks and sediments to animal shells and tubes. These thin layers of microbes create localized zones of enhanced nutrition and chemical activity. Grazers often target these films directly, harvesting microbial biomass in a world where plants do not grow. At this scale, life feeds on life in its smallest, most persistent form. In deep sediments, microbial communities organize into layers based on oxygen availability. Near the surface, oxygen allows certain bacteria to dominate, while deeper layers favor anaerobic microbes that use alternative chemical pathways. These vertical gradients structure entire benthic ecosystems. Animals that burrow or feed at specific depths interact directly with these microbial zones, linking microscopic chemistry to macroscopic behavior. Enzymes adapted to high pressure and low temperature enable biochemical reactions to proceed efficiently in deep environments. These enzymes often have flexible structures that resist pressure-induced distortion. Without such adaptations, basic metabolic processes would slow or fail. At the molecular level, survival in darkness depends on chemistry that works reliably under extreme physical conditions. Cell membranes in deep-sea organisms contain specialized lipid compositions that maintain proper fluidity under pressure. These membranes allow nutrients, signals, and waste products to move efficiently despite crushing forces. This adaptation is subtle but essential. Without it, cells would lose integrity, and life at depth would be impossible regardless of other adaptations. Benthic biomass at depth. Their shells contribute to sediment formation, and their feeding helps regulate microbial populations. At this scale, even single-celled organisms play roles that ripple upward through the ecosystem. Radiolarians drift through dark waters, their silica skeletons acting as both feeding structures and eventual contributors to deep-sea sediments. They capture tiny prey and particles, sustaining themselves without any reliance on light. When they die, their skeletons sink, linking the water column to the sea floor. Over time, this microscopic range shapes sediment chemistry and structure. Copepods remain abundant even in aphertic zones, feeding on particles, microbes, and smaller plankton. Their small size and efficient reproduction allow them to exploit diffuse energy sources. They form a crucial bridge between microbial production and larger consumers, transferring energy upward through the food web in tiny, steady increments. Ostracods, minute crustaceans, contribute to deep-sea ecosystems through feeding and, in some species, bioluminescent signaling. At close range, their light emissions can serve communication or defensive functions. Even at this scale, light becomes meaningful in darkness. Small flashes carry information that can influence survival and reproduction. Chemical gradients at the microscale guide behavior in animals that will never see light. Dissolved compounds diffuse through water and sediment, forming invisible trails that indicate food or danger. Following these gradients requires extreme sensitivity, allowing organisms to orient themselves in a world defined by chemistry rather than sight. Microbial respiration consumes oxygen in sediments and water, shaping where animals can live. Oxygen depletion creates boundaries that influence burrowing depth and feeding behavior. These invisible limits structure ecosystems as effectively as physical barriers, all operating silently in the dark and influencing microbial population dynamics. Through infection and cell lysis, they release organic matter back into the environment. This viral loop enhances recycling, ensuring that energy continues to circulate even at the smallest scales. In darkness, nothing is wasted. Nanoparticles of organic matter can be taken up directly by some microbes, bypassing larger food web steps. This allows energy to move efficiently, even when particles are extremely small. Such pathways emphasize that deep ecosystems rely on flexibility and redundancy at microscopic levels. Mineral particles interact with organic molecules, affecting how nutrients are preserved or released. Clay minerals can bind organic compounds, slowing decomposition. This interaction influences long-term carbon storage and availability, connecting microscopic chemistry to global cycles. At hydrothermal vents and seeps, microbial mats form dense layers fueled by chemical energy. These mats are the primary producers of submerged ecosystems. Grazers feed directly on these microbes, transferring chemically derived energy into animal biomass at the smallest possible scale. Even sound and vibration interact with microscopic particles, subtly influencing aggregation and dispersion. Physical forces shape biological processes continuously. In the deep ocean, physics, chemistry, and biology are inseparable at these scales. All of these microscopic processes operate quietly, endlessly, and without light. Together, they form the slow heartbeat of the deep sea. From particles to cells, from enzymes to gradients, life persists gently, one small interaction at a time. In the deep ocean, understanding life that has never seen light depends on careful observation, indirect measurement, and technologies designed to work gently in dark, extreme conditions. because darkness, pressure, and distance limit direct contact, much of what is known comes from patient detection rather than constant observation. These experimental and measurement-based approaches quietly reveal how deep-sea systems function, one careful signal at a time. These vehicles move slowly to avoid disturbing animals adapted to stillness. The lights used on ROVs must be carefully managed, as sudden illumination can alter behavior. Observations gathered this way capture rare interactions, postures, and movements that cannot be inferred from collected specimens alone. Autonomous underwater vehicles expand exploration by operating without direct human control, mapping large areas of dark seafloor over extended periods. These systems collect data on temperature, chemistry, topography, and biological presence in regions too vast for single dives. By returning repeatedly to the same locations, they reveal slow changes in deep ecosystems, offering insight into processes that unfold over years rather than dapper moments. Deep-sea trawls and traps provide physical samples, though they represent only brief snapshots of life at depth. Many fragile organisms are damaged during collection, highlighting the limits of these methods. Even so, samples allow laboratory analysis of anatomy, genetics, and chemistry, connecting physical structure to ecological role in environments never touched by sunlight. Environmental DNA sampling detects the presence of organisms through genetic material suspended in water or sediment. This technique allows identification of species without seeing or capturing them directly. In darkness, where observation is difficult, environmental DNA expands knowledge of biodiversity, revealing hidden species and distributions across deep habitats. Acoustic monitoring records sounds produced by geological activity, water movement, and sometimes by animals themselves. Because sound travels efficiently in water, it provides information across long distances in the absence of light. These acoustic signals help map habitat features and detect events such as landslides or feeding activity that would otherwise go unnoticed. Chemical sensors measure dissolved gases, nutrients, and compounds that signal biological or geological processes. Changes in chemistry can indicate the presence of vents, seeps, or decaying organic matter. In the deep sea, chemical shifts often provide the first clue that life is active nearby, guiding further investigation. Pressure-resistant cameras are designed to function reliably at extreme depths, capturing images under crushing forces. These devices allow visual documentation of life forms adapted to permanent darkness. Each image represents a careful balance between illumination and intrusion, preserving natural behavior while revealing unseen worlds or conditions. These systems operate quietly, observing without interference. Such long-term records reveal patterns of movement, feeding, and community change that short visits cannot detect. Stable isotope analysis traces the sources of energy in deep ecosystems by examining chemical signatures in tissues. This method reveals whether organisms rely on surface-derived organic matter or chemosynthetic production. These measurements clarify food web structure in environments where direct observation of feeding is rare. Respirometry experiments measure oxygen consumption to estimate metabolic rates of deep-sea organisms. These experiments must account for pressure and temperature to remain accurate. Results show how metabolism is slowed and optimized in darkness, reflecting energy conservation strategies. Genomic sequencing uncovers genetic adaptations that support life without light, including pressure tolerance and sensory specialization. Comparing genomes across depths reveals evolutionary responses to darkness. These studies connect molecular changes to ecological function, bridging laboratory analysis and deep-sea reality. In situ experimentation places instruments directly in deep habitats to observe biological responses under natural conditions. This avoids artifacts caused by bringing organisms to the surface. Such experiments preserve the integrity of behaviors shaped by darkness and pressure. Optical sensors detect bioluminescent flashes, recording timing, intensity, and frequency. These measurements reveal communication patterns invisible to the human eye without instrumentation. In the deep sea, tracking light emissions is often the only way to observe interaction at a distance. Sediment cores provide layered records of past biological activity, capturing organic content and microfossils deposited over time. These cores allow reconstruction of historical deep-sea conditions. Through them, scientists infer how dark ecosystems responded to past climate changes and surface productivity shifts. Tracer experiments introduce harmless markers to follow water movement and particle transport. These reveal how food and chemicals travel through dark waters. Understanding these pathways explains how distant processes influence deep communities. Physiological sensors attached to animals record movement, depth, and sometimes heart rate, offering insight into behavior in darkness. These devices must be unobtrusive to avoid altering natural activity. Laboratory pressure chambers recreate deep-sea conditions, allowing controlled experiments on physiology and behavior. These chambers simulate darkness, cold, and pressure. They help identify which traits are fixed adaptations and which are flexible responses. Photometric instruments measure how light propagates and is absorbed in deep water. These measurements inform understanding of visual ecology and camouflage. They clarify why certain colors and skin structures evolved in lightless environments. Biogeochemical sensors monitor nutrient cycling, revealing how elements move through deep ecosystems. These measurements connect microbial activity to larger-scale processes. They show how dark ecosystems participate in global cycles despite their isolation. Deep-sea observatories establish permanent monitoring stations on the seafloor. These installations collect continuous data on environmental and biological variables. Over time, they reveal trends and rare events otherwise missed. All of these experimental and detection-based methods work together to illuminate life in darkness without breaking it. Each tool adds a quiet piece to a vast puzzle. Through patience and precision, science listens to the deep sea rather than forcing it to speak. In the deep ocean, biology turns inward, refining bodies and senses to function in a world without light. Physiology, perception, and internal balance become the primary tools for survival, shaping animals whose entire experience unfolds in darkness. These adaptations are not dramatic bursts of change, but quiet refinements that allow life to continue steadily under pressure, cold, and silence. Possess eyes designed to capture the smallest possible number of photons, prioritizing detection over detail. In environments where bioluminescence provides the only light, being able to notice a faint glow can matter more than forming a sharp image. This leads to eyes with large pupils, reflective layers, or tubular shapes that focus on narrow fields of view. Vision becomes a specialized sense, not for seeing landscapes, but for recognizing brief, meaningful signals in the dark. In species where light is too rare to be useful, eyes may shrink, simplify, or disappear entirely. Maintaining complex visual systems requires energy, and in permanent darkness, that energy can be redirected toward other sensory pathways. These animals rely more heavily on smell, touch, and vibration detection, demonstrating that losing a sense can be an adaptive gain when the environment no chas longer supports its use. Chemoreception is often highly developed in deep-sea organisms, allowing them to detect minute concentrations of dissolved substances. This sensitivity enables animals to follow chemical trails from food sources, mates, or decaying matter across long distances. In darkness, smell becomes a map. Chemical cues replace visual landmarks, guiding movement through invisible gradients that shift slowly with currents. Mechanoception, the ability to sense movement and vibration in water, is another critical sense. Many deep-sea fishes and invertebrates detect pressure waves caused by nearby motion, allowing them to sense approaching prey or predators without seeing them. This sense creates an awareness of space defined by motion rather than by sight, where even subtle disturbances carry information in otherwise still water. Bioluminescent organs are integrated into physiology with precise control. Light production is often regulated by nerves or hormones, allowing animals to emit flashes only when needed. This control prevents wasted energy and reduces unwanted attention. In the deep sea, light is not a constant glow, but a carefully timed message. Some species possess photophores arranged in specific patterns, embedded into skin or along the body. These patterns may serve roles in species recognition or alignment within groups, functioning like identifiers in a world where color and shape are difficult to perceive. Physiologically, these light organs are part of the body's communication system, linking internal state to external signaling, allowing organisms to survive long periods between meals. This slow pace is not weakness, but balance. Physiology matches environment, ensuring that intake and expenditure remain aligned over long spans of time. Many deep-sea animals have soft, gelatinous tissues that reduce the energetic cost of building and maintaining rigid structures. These tissues also compress easily under pressure, reducing mechanical stress on the body. Physiologically, softness becomes strength, allowing survival in conditions where stiffness would be costly or damaging. Pressure tolerance is embedded deeply into cellular function. Proteins, enzymes, and membranes are structured to maintain their shape and activity under immense force, preventing collapse or malfunction. These molecular adaptations are invisible but essential, supporting every other physiological process in a lightless, high-pressure world. Oxygen use is carefully managed in deep-sea species, especially in regions where oxygen levels are low. Some animals have blood proteins with high oxygen affinity, allowing them to extract and transport oxygen efficiently. This adaptation supports survival in environments where oxygen is scarce and must be conserved as carefully as food. Digestive systems are often designed to maximize extraction from limited resources. Some animals digest slowly, allowing thorough absorption of nutrients from low-quality food. Physiology here emphasizes completeness over speed, ensuring that little is wasted in a world where replacement is uncertain. Reproductive physiology reflects the challenges of darkness and distance. Gametes may remain viable for extended periods, increasing the chance of fertilization when encounters are rare. This internal timing replaces external cues like daylight or seasons, allowing reproduction to proceed independently of light-based rhythms. Some species invest heavily in developing fewer offspring, providing them with large energy reserves or prolonged protection. This physiological investment increases survival odds in environments where suitable conditions may be rare. The body becomes a buffer against environmental uncertainty. Sensory integration in deep-sea animals often combines multiple inputs, chemical, mechanical, and sometimes visual, into a unified perception of kelor. Surroundings. No single sense dominates completely, most reliable information available in darkness. Bioluminescent detection is often wavelength-specific, tuned to the most common emissions in deep water. Retinas may be structured to maximize sensitivity to blue-green light. This specialization reflects physiological fine-tuning to the actual light environment, rather than to an abstract concept of vision. Some predators possess specialized jaws and muscles that remain effective at low temperatures. Muscle fibers are adapted to contract efficiently in cold, preventing sluggish responses. This ensures that when an opportunity arises, the body can respond decisively, despite the slow background pace of life. Nervous systems in deep-sea animals often prioritize reliability over speed. Signal transmission may be slower, but it remains consistent and energy efficient. This steady processing suits environments where rapid reaction is less important than accurate interpretation of rare events. Circadian rhythms tied to day and night are often reduced or absent. Instead, internal clocks may respond to feeding cycles, pressure changes, or chemical cues. Physiology becomes decoupled from the sun, operating on rhythms defined entirely by the deep environment. Overall, deep-sea biology reflects a quiet harmony between body and world. Each physiological trait is shaped by darkness, pressure, and time. Life does not fight the dark here. It settles into it, adjusts gently, and continues. In the deep ocean, behavior unfolds slowly, often at scales measured in years rather than moments. Without sunlight to drive daily rhythms or seasonal change, life proceeds according to long internal clocks, subtle environmental shifts, and rare events that leave lasting impressions. These behaviors are quiet, patient, and persistent, shaped by time more than urgency. Many deep-sea animals spend long periods almost completely still, conserving energy in environments where movement offers little immediate reward. This stillness is not inactivity, but a deliberate behavioral state that reduces detection and preserves internal reserves. Episodic. Animals may go long stretches without eating, then feed intensively when a food source appears, adjusting digestion and metabolism accordingly. This feast-to-famine rhythm is a behavioral adaptation to unpredictable input, allowing survival without constant access to nourishment. Scavengers demonstrate particularly patient behaviors, slowly following chemical trails across great distances toward decaying matter. Their movements are guided by faint signals that persist over time, rather than by immediate visual confirmation. This slow pursuit reflects confidence in chemistry and endurance, trusting that the signal will eventually lead to reward. Predatory behavior often involves long-term positioning rather than active searching. Predators may select locations where prey is likely to pass, remaining there for extended periods. Such ambush strategies reduce energy use and increase success when encounters are rare, making patience a defining behavioral trait. Social interactions in the deep sea are typically limited and subtle. Encounters between individuals may be infrequent, and behaviors such as courtship or signaling are often brief and highly efficient. This minimizes exposure and energy cost, while still allowing reproduction and species recognition in darkness. Bioluminescent behaviors are often carefully restrained. Many animals emit light only under specific circumstances, such as defense, attraction, or communication, avoiding unnecessary signaling. Behavioral control over light use reflects an understanding, built by evolution, that elimination carries risk as well as benefit. Migration in deep-sea animals can occur over long timescales, driven by changes in food availability or developmental stage, rather than by light paths. Cycles. These movements are often vertical, spanning hundreds or thousands of meters. Such migrations unfold slowly, guided by internal cues and environmental gradients, rather than by visual landmarks. Reproductive timing in the deep sea may be spread over long periods rather than concentrated into short seasons. Some species release gametes intermittently, increasing the chance that at least some will encounter compatible partners. This behavior reflects adaptation to sparse populations and low encounter rates. Burrowing behavior is often slow and methodical, with animals maintaining and reusing tunnels over long durations. These burrows provide stability and protection in an otherwise exposed environment. Maintenance of such structures becomes a long-term behavioral investment rather than a temporary shelter. Growth-related behavior is similarly extended. Juveniles may take many years to reach maturity, adjusting feeding and movement gradually as their needs change. This slow progression aligns with long lifespans and stable environmental conditions. Avoidance behavior in the deep sea tends to be subtle. Rather than fleeing rapidly, animals may simply reduce movement or alter orientation to minimize detection. In darkness, invisibility can be more effective than escape. Territoriality, where it exists, is often weak and loosely defined. Energy constraints make constant defense impractical, favoring tolerance and spacing instead. Behavior focuses on coexistence rather than confrontation. Some deep-sea species exhibit site fidelity, returning repeatedly to the same locations over long periods. Familiarity with chemical and physical features can provide reliable access to food or shelter. This long-term association with place replaces the need for visual mapping. Parental behaviors, when present, can extend over long durations. Guarding eggs or young may involve weeks or months of low-activity vigilance. Such care reflects a trade-off between mobility and offspring survival. Response to disturbance is often slow. After events such as sediment shifts or food falls, behavioral changes may unfold gradually as animals assess altered conditions. Recovery behaviors emphasize adjustment rather than rapid recolonization. Learning, where it occurs, is likely based on repeated chemical and mechanical experiences rather than visual memory. Familiar cues become associated with safety or opportunity over time. Behavioral memory in darkness is built from patterns, not pictures. Interactions between species often rely on indirect effects rather than direct encounters. One species feeding may alter chemical conditions that influence others long afterward, restrained and aligned with long-term survival rather than short-term gain. Time itself becomes the dominant behavioral factor. In the deepest parts of the ocean, life settles into its quietest expressions. Here, change is slow, motion is gentle, and existence unfolds without urgency. These final reflections drift through the calmest layers of deep-sea life, where time stretches and darkness feels steady rather than empty. Life continues not through effort, but through balance. Many deep-sea organisms exist in a state of near-constant equilibrium, where energy intake and expenditure are carefully matched. Their bodies and behaviors are tuned so precisely to their environment that long periods pass without noticeable change. This balance allows them to persist calmly without the need for rapid adjustment in a world where conditions remain stable and predictable. Silence dominates the deep ocean, broken only by faint currents or distant geological sounds. Animals living here are adapted to this acoustic stillness, where sudden noise can be more disruptive than helpful. In such an environment, quiet itself becomes part of survival, encouraging low movement and restrained interaction. Darkness in the deep sea is not experienced as absence, but as constancy. There is no transition from light to dark, no anticipation of dawn or dusk. This constancy removes urgency from life, allowing organisms to exist without daily cycles or visual expectations. Many deep-sea animals show behaviors that appear almost meditative, drifting slowly or remaining still for long durations. This is not inactivity, but an efficient response to an environment where haste offers little advantage. Stillness conserves energy and blends seamlessly with the surrounding water, becoming a natural state rather than a pause. Growth in the deep ocean is often imperceptible over short timescales. Organisms add tissue slowly, spreading development across years or decades. This gentle pace aligns with stable conditions, allowing life to unfold without pressure to hurry. Reproduction, too, can be quiet and prolonged. Instead of dramatic seasonal events, some species reproduce gradually, releasing offspring or gametes over extended periods. This approach softens the risks of failure, letting chance and time work together. Death in the deep sea is rarely sudden or dramatic. When it occurs, it often becomes part of a slow transformation, feeding microbes, scavengers, and sediments. Nothing is wasted. Each ending gently becomes a beginning for something else. Deep-sea communities often persist in roughly the same form for long spans of time. Without frequent disturbance, relationships between species stabilize. This stability fosters continuity rather than change, favoring endurance over innovation. The absence of light also means the absence of visual spectacle. There are no colors to display, no patterns meant to be seen from afar. Beauty here exists in function, balance, and subtle interaction rather than appearance. Many deep-sea animals appear indifferent to time, as it is measured at the surface. Without seasons or days, their internal rhythms stretch and soften. Life becomes guided by internal needs rather than external clocks. In these environments, patience is not a strategy, but a condition of existence. Everything unfolds at its own pace, unforced and unhurried. Survival depends on acceptance rather than resistance. Deep-sea ecosystems remind us that abundance is not required for persistence. Even sparse resources, when used efficiently, can sustain life indefinitely. Scarcity becomes manageable when time is abundant. The deep ocean also teaches that complexity does not always require speed or competition. Cooperation, recycling, and restraint can support rich systems. These systems thrive quietly, without display. There is no rush to evolve quickly in such stable darkness. Change happens slowly, layered gently over generations. Evolution here is patient, refining rather than reinventing. Life without light shows that awareness does not require vision. Sensing, responding, and existing can occur through other means. Perception adapts to what is available. The deep sea holds no preference for novelty or drama. It favors continuity, subtlety, and quiet persistence. Its creatures reflect this calm. In this world, time feels wide and forgiving. Moments stretch, and life has space to simply continue. Nothing demands attention. As we drift away from these depths, the quiet remains behind, unchanged by observation. Life there goes on as it always has, steady, gentle, and unlit. And so, in the deep ocean's permanent night, existence does not sleep, but it does rest. Here, everything moves slowly, and nothing needs to hurry.